So we'll be learning about hookworm infections today. So let's say what happens to you is a patient presents to you with symptoms of asthma, but he's also having diarrhea. Now, asthma, you can say it could be due to type 1 hypersensitivity reaction due to protected course of life, but then diarrhea could be bacteriogenic. Then what you do is you're going to do some tests, blood tests, and in which you're going to see sign of failure. So you start to suspect worm infestation, and can that's when we can get a differential diagnosis of hookworm, which we're going to study today. So hookworm classification. Hookworm is of hookworm hookworm classification, that is parasitology. So hookworm infection is widespread in the world, and it is also seen in India. Uh, Sorry. There are two species are human pathogens that cause intestinal disease. Number one is going to be Ancylostoma duodenale, and number two is going to be Necator americanus. Ancylostoma duodenale is the old world hookworm, whereas Necator americanus is the new world hookworm. Then there's also animal parasites, which are cutaneous larva migrants, such as Ancylostoma brasiliensis, Ancylostoma caninum, and Ancylostoma selanicum. Then is the epidemiology. So hookworm infestation or infection occurs to more than 900 people in this world, out of which 835 is caused by a Necator and 135 is caused by Ankylostoma. Whereas in India also it is predominantly C, so 200 million people are affected, and these are heavily infected in areas like Assam due to tea plantation, West Bengal due to the same reason, Bihar, Odisha, Andhra Pradesh, Tamil Nadu, Kerala, and Maharashtra. Now, in North India, it is going to be more predominant, and South India is also predominant. So, age and sex. So, it affects mainly males and young adults. But when it occurs in children and pregnant women, the anemia can be severe. Then is the endemic index, that is also known as the Chandler's index, which helps you to estimate the morbidity and mortality by warm load. There's also one more thing that UP and Punjab, uh, you can't find Necator over there, so Necator is going to be absent in UP and Punjab. How does the hookworm look? So this is the morphology, or so there are going to be three forms, three morphological forms, like all the other nematodes. So number one, there's going to be an egg. Now, egg cannot be differentiated with the ones from the strongyloids. Then there's larva, that is L1 larva, L2 larva, L3 larva, and L4 larva. Out of which L1 larva is known as rhabditiform larva, and L2 larva is known as filariform larva. Then is the adult worm, that is 7 to 13 millimeter, and it is going to be bent anteriorly, and that is why hookworm is known as is given this name, because this kind of looks like a hook. Then how do you then this is our adult worm. So this is going to be Ankylostoma duodenale, and this is going to be a Necator americanus, and this is how we differentiate them. So over here, buccal capsule is going to have teeth, whereas for Necator americanus, the buccal capsule would have cutting plates. And there's larval stages. So there's L1 and L3 stages, which are the main stages that help us to diagnose the infection. So number one is L1, that is the rhabditiform stage. So this is the stage that can sometimes be detected inside the stool, but it is very difficult to differentiate which larva is which. That is, we don't know whether it is caused by Necator americanus or Ankylostoma duodenale or strongyloids due to the appearance of L1 larva. That is when we're gonna wait for it to grow, and we're gonna see the L3 filariform larva. Uh, what is the life cycle? So this is the life cycle. Number one is when a person walks on a damp soil, whenever he walks barefoot, as we've discussed in Assam and tea plantations, etc. The person, the larva, that is the L3 larva, that is also known as the filariform larva, it is gonna penetrate the skin of the person. So after which it would enter the subcutaneous venules, from which it is gonna travel towards the lungs. Now, in the lungs, it is going to travel upwards, and it is going to go to the trachea. Through the trachea, it is going to go into the pharynx, and from which it is swallowed. Now, after swallowing, it is going to go into the intestine, and over there, the intestinal phase starts. So, L3 larva converts into L4 larva, and L4 larva converts into adult form, adult worm, after molting twice. Now, this adult worm is attached to the intestinal mucosa with its teeth, and then these adult worms would lay eggs, and these eggs are going to be non-embryonated, unembryonated, and these are the diagnostic form, and these can be detected inside the feces. Then these unembryonated eggs would convert into embryonated eggs inside the soil, and they're going to hatch and form L1 larva, which would molt into L2 larva, and which then forms the filariform larva, that is the L3 larva. And this filariform larva then penetrates into the skin, and this would continue the cycle of infection that we've known. Then is going to be what is the pathogenesis. So hookworm has the this ability to suck blood from the intestinal vessels. How? Number one is by attaching and making cuts in the intestinal wall by buccal capsule and teeth, followed by sucking the blood. And number two is it is going to secrete hydrolytic enzymes and anticoagulants, due to which the blood does not clot, and this makes sure that there's a continuous flow or oozing of blood from the attachment site. Now, what is the amount of blood that is taken? So, the blood loss in ankylostoma is going to be 0.15 to 0.26 ml per worm per day, and in the case of Necator americanus, it is going to be 0.03 ml per worm per day. Now, what are the clinical manifestations? So number one is going to be the cutaneous lesions. So wherever at the site of penetration of the larva, site of penetration of the larva, you're going to see a small itchy maculopapular rash at the site of the skin penetration. So it is going to be pruritic, that is itchy. Number two is you're going to see serpiginous tracts, that are snake-like tracts from wherever the larva has taken its root. So which would be subcutaneously. So you're going to see subcutaneous serpiginous tracts. Then you're going to see L3 larvae. It is going to promote dermatitis and tracts. So tracts are going to be formed, and since the larva, which is a foreign substance, is passing through your skin, it is going to provoke dermatitis. Now, what happens is after passing through the venules of the legs, it is going to go towards the lungs. When it goes towards the lungs, it is going to cause mild transient pneumonitis. So, migrating L3 larva inside the lung, they're going to cause some symptoms such as pneumonitis, asthma, bronchitis. And these symptoms are going to be less severe as compared to those of ascariasis. The migrating larva severity is going to be more in the case of ascaris as compared to hookworm. Then are the manifestations when the worm reaches inside the intestine. So number one is going to be asymptomatic. So this is seen in most of the infections. So this is when the worm load is going to be low. Then there are early intestinal manifestations when the worm load is kind of moderate. So you're going to see epigastric pain inside the person, you're going to see inflammatory diarrhea, and there's going to be peripheral eosinophilia in the blood tests. Then after a lot of time, when the infestation by worm it's chronic and the worm load is going to be heavy, that's when you're going to have the late intestinal phase. So what is going to be seen in this? Number one is going to be iron deficiency anemia. So the worm is slowly taking away blood from the person, and it is depriving the person of its nutrients, due to which he's going to have iron deficiency anemia. Also, the worm is going to deprive the person of more nutrients and proteins, due to which the person is going to have malnutrition, is going to have weakness, is going to have dyspnea, and also dyspnea due to iron deficiency anemia. And since the important nutrients are impaired, and this is mostly seen in the case of children, the person would have impaired intellect. Then there's something known as Wakana's disease that is also caused by hookworms. That is when Ancylostoma duodenale is ingested orally. So you're gonna see L3 larva seen infested orally. So the person presents as nausea, vomiting, cough, dyspnea, and pharyngeal irritation. But this is not seen with Necator americanus. So clinical spectrum depends upon the worm load. If the worm load is very low, the person is going to be asymptomatic, and if it is very high, they're going to suffer from chronic malnutrition and anemias. How do you diagnose the worm infection? So you're going to do stool microscopy. First, you're going to collect stool from the patient in a sterile screw-capped container. After which, you're going to prepare a fresh mount, and you're going to look at it under the microscope, where you're going to see characteristic eggs that are found in fresh feces. Now, these eggs may sometimes hatch in order to form rhabditiform larva, and it is very important to differentiate it with strongylides, which already have larvae inside their feces. Now, this is now we're going to check for the characteristics of hookworm eggs. So hookworm eggs is going to have a thin eggshell. It is going to have a thin eggshell, as you can see over here. And inside the egg, there are going to be segmented ovum that is known as blastomeres. So there's typically four blastomeres, which I think is represented inside the picture. So it would typically contain only four blastomeres, and there is going to be the presence of a clear space around this segmented ovum. And when you look at it under the microscope, you're going to see that it does not get stained by bile. So you're going to color it with it is going to be colorless under a saline mount, and it is not bile stained, and the size is going to be 60 to 40, 60 by 40 micrometer. So it is going to float in saturated salt solution when you're going to do egg concentration techniques in order to increase the burden of parasite. And eggs of Ancylostoma duodenale and Necator americanus are indistinguishable. Now, in order to count the eggs and to check the disease burden, there's something known as the CATO-CADS technique, and in which you can measure eggs per gram of stool, which helps us to determine the intensity and morbidity of the disease. So, uh, the next thing that you're going to do is you're going to do stool culture. So, in case you didn't find any larvae or eggs, you're going to culture it in order to have better results, or to find out uh, which parasite it is. So you're going to do something known as Harada Mori filter paper tube method, which helps you, in which you're going to wait until the eggs inside the fresh tool are going to hatch and form the L3 filariform larva in 5 to 7 days. And there's also known as the Bierman funnel technique, which also works on the same principle. And there's something known as agar plate technique, which is more sensitive. And after the L3 larva hatches, that is when you can differentiate Ancylostoma duodenale from Necator americanus. Now, molecular diagnosis is you can do PCR. PCR would help you to detect a gene inside, amplify a gene and detect it, which would be specific for that particular parasite. Now, in this case, it would target mitochondrial cytochrome oxidase, and it is species specific, that is Ancylostoma versus Necator. And this technique can help you to differentiate between Ancylostoma and Necator, and it's going to be highly sensitive, that is 1 copy per 200 milligram of stool. Now, other findings that you're going to see are you're going to see eosinophilia, peripheral eosinophilia inside the person's blood. The person would have hypoalbuminemia, albuminemia inside, and the person will also suffer from hypochromic microcytic anemia secondary to iron deficiency. Then this is the stool microscopy. So this is the typical egg of Ancylostoma duodenale, in which there is going to be thin outer shell, a clear space in between, and there is the presence of four blastomeres inside the stool of the patient of the egg. Then this is the brief idea of lab diagnosis. So there's going to be primary screening and specimen selection, collection. Then there's going to be direct wet microscopy. There's going to be stool culture by Harada Mori and agar plate technique. Then you're going to do morphological identification by looking at the number of blastomeres and the clear space and non-biostain egg. That you're going to see for filariform larva examination in order to distinguish it from the Necator, to distinguish Necator from Ancylostoma. You're going to do advanced text testing, such as PCR, in which you're going to detect mitochondrial cytochrome oxidase, and you're going to see for supportive evidence that is hematological, such as microcytic hypochromic anemia, peripheral blood eosinophilia, and hypoalbuminemia. Then we're going to see for Chandler's index, which is used to determine the morbidity and mortality based on the average egg count. Then this is how you differentiate strongylides from rhabditiform larva of Ancylostoma or Necator. Um, this is what is the treatment? So treatment is going to be you're going to give an anti-parasitic. So mainly albendazole 400 mg is given, or mebendazole 500 mg are the primary first line of choice for this. You can also give parental pamoate. But recently, efficacy and resistance have been reported for albendazole and mebendazole. Then, apart from giving or killing the worm by albendazole or mebendazole, you're also going to give symptomatic treatment. That is going to be for mild anemia in the case of or oral iron and folic acid. And you're also going to treat the severe diseases that it has caused, such as in the case of severe disease, you're going to treat protein loss and malabsorption by giving parenteral iron replacement and nutritional support. How are you going to prevent this is improve personal hygiene always. You can wear shoes when working in such areas. You should properly dispose the feces so that the fecal-oral contamination to other people can stop. You have to improve your nutrition, such as increase the dietary intake of iron and treat whatever infected persons are there. The last thing is soil transmitted helminth infections. So, helminths that are transmitted by soil, these affect more than 1.5 billion people in this world. That is about 24% of global population. So, this is seen, these infections are caused by Ascaris, Trichuris, and Hookworm, and these are transmitted through contaminated soil. Now, these are seen in Africa, America, China, and East Asia. So what are the high risk groups? So number one is going to be preschool children, then school age children, pregnant women, and the adults in high risk occupations such as tea pickers and miners. Now, why is this important? Is because in school age children and sometimes in preschool children, they have a lot developing in their body. So due to worm infestations, their intellect can be impaired, and they can also be physically impaired due to malabsorption, anemia, diarrhea, and dysentery. So, what is the solution to this? There is a day that is known as deworming day, that is on 10th Feb and 10th August, which are the natural deworming days in India. So, what you're going to take a single dose of albendazole, that is 400 mg for ages 1 to 19 in order to prevent worm infestations. So, WHO aims to eliminate morbidity by 2030 and target is less than 2% moderate or heavy infections. Thank you.